Good afternoon, everyone. Thanks for being here still at the end of the meeting. I have the dubious pleasure of being the last speaker. I'll be talking to you today about some historical data within the human microbiome field and some of the ideas that actually translate to animal work. And then I'll finish talking a little bit about the data that we're gathering in Brazil in my lab. Our interests have always been, and just in the matter of interest, I have no conflict of interest to declare. Since I started working with the gut microbiome, and you guys need no introduction to what actually it does, it became very curious of how it actually is playing around with populations and how it's actually changing or presenting itself and regulating health. Over the years, we have come to see that we actually are talking about co-evolution, co-development, co-metabolism, and co-regulation of our physiological processes. So it's a very, very close relationship with all vertebrates. Probably all eukaryotic cells, since they became eukaryotic cells, have dealt with prokaryotes. And we decided at some point, as a medical field overall, that we would like to use this as a source to actually track patterns within our physiology, as if they were separate. But what we can see is that there is a series of relationships that actually can be replicated across different cohorts, very large cohorts, that actually represent things that are happening within our metabolism, and we can actually detect relationships with the gut microbiome composition. So we have some examples in the board from two very large cohorts, and these studies have been replicated over time, and some of them have become actually clinical markers that we can actually use. And this is one of the great advantages of actually using the microbiome, particularly fecal microbiome, because it's non-invasive, and we can actually get a poop sample and do something with that relatively quickly today, and take action on top of it. Over the years, we actually have moved away from just clinical parameters to actually trying to understand at the molecular genetic level. We know now epigenetic regulation, we know genetic regulation, and we have a fine understanding of this relationship of the bacteria with several of those physiological processes. The knowledge has actually become so refined that products are actually being developed to target directly the bacteria within the gut, and we have some trials already being produced, for example, with the TMAO story, right? So it starts with choline and those trimethylamine, which get converted by the liver in TMAO. And if you inhibit the enzyme within the bacteria population within the gut that actually does this initial conversion, you remove that from circulation. And that doesn't need to be absorbed. That doesn't need to be dealt within the context of liver metabolism, for example, as a drug. So it has a lot of potential to actually act before it becomes a problem within the host. So the potential for the microbiome actually is, I think, very unique. And with that said, it's a very, very ancient relationship. We can actually backtrack the relationships that we have across all mammals and across all vertebrates, actually, and recapitulate those relationships just looking at bacteria within the gut. So studies have already shown this, that if you just take the bacteria within the gut and use that to make a pseudo-phylogeny of those groups, you actually reproduce that quite well. We can see that a little bit more zoomed in, for example, with primates. What we have on the left is actually genetic markers to actually make the phylogeny of humans, chimps, and gorillas. And you can see that the pattern is exactly the same if you just look at the bacteria within the gut. So this relationship is really, really old. And we are at a point where we can actually integrate this genetic and environmental relationships that are guiding the gut microbiome composition. Very early on, it became very obvious that we have some very strong effects of the types of diet. This study was one of the first ones that actually looked at the very, very large types of mammals. And what we can see is the very strong relationship of the type of diet that these animals have. So whether they're herbivores, carnivores, or omnivores, and you can see that in the left panel. But we can also recapitulate to some extent also the segregation of the different taxa, right? So again, a very strong and ancient relationship between these two groups. As animals and as people, we are not isolated entities. We actually live together. So all changes that are happening within the human population are also being reflected throughout the world within all animals, including the ones that live with us. And and I think this picture is amazing because it was the first time that we actually saw how intricate is this relationship of microbiome between pets and people. And I'll point just a couple of things within the PCOA plot here. Everybody probably remembers this. If the points are close to each other, that means that the microbiome is very similar to each other. And what we see here is that the microbiome in green from the tongue of dogs is actually very similar to the microbiome from the skin of people in yellow, on the hands of those people. So it sort of makes sense, right? If you actually own a dog, that your hands are very similar in bacterial composition to what you find on the dog's saliva. And this exchange of bacteria is happening actively throughout time and throughout our lives with the environment and between species. Some of it is controlled by what we gain from the environment. We're colonized from birth. And some of it is actually actively maintained and stimulated by diet. Diet is probably the strongest effect that we have in controlling and maintaining the gut microbiome. This was shown very nicely very early on when people started looking at the gut microbiome, comparing populations that have very distinct diets, that you had very distinct patterns within the gut of those populations. So in this seminal study, we saw that people from Burkina Faso and people from Italy had very, very distinct gut compositions. And this was one of the first papers that actually suggested and had some indirect evidence for this relationship with diet. Shortly after that, we saw that we can actually segregate humans just based on enterotypes. The term is very controversial to this date. Some people love, some people hate it. But it's a very useful tool to actually find distinct clusters of people that actually are more similar to each other. Whenever you do this enterotyping technique, and you can do that in animals as well, you can find those clusters. And this is all healthy people, right? You don't need to do this in disease. You can actually see this in healthy people. Two of those clusters are actually very, very much related to the types of diets that we have as humans. So the Bacteroides component of the microbiome and the Prevotella component, they're two genera of the gut microbiome in humans. They're very much work as a trade-off from each other. Pretty much everybody has Bacteroides, but Prevotella within Western industrialized societies seems to be more occasional. And they're very much driven by the intake of carbohydrates and proteins. So a very, very rich diet in meat, the churrasco of South America, for example, drives a microbiome that's actually very rich in Bacteroides. Whereas people that tend to have carbohydrate-rich diets actually have a microbiome that is actually very rich in Prevotella. This story was actually presented and done and discovered and repeated and confirmed in industrialized countries. And when we step outside of this comfort zone, we started to see that this is not necessarily the same for other populations around the world. So we see, for example, that if you take people from Thailand, actually live and grew up and live in Thailand, take their children and people that are immigrating to the US, and then compare to the children that actually are born in the US, grown up in the US, but actually are sons of Thai immigrants, you see shifts in this microbiome pattern towards the pattern that's normally present within an European descent people of many generations within the US. And the longer you spend in the US, the closer you reach to this level and this balance between Bacteroides and Prevotella, enriching the amount of Bacteroides on the intestine and losing the Prevotella from the intestine. So every time that we step outside of this industrialized, urbanized scenario, we started to see that we actually have shifts in the gut microbiome to a pattern that some people have dubbed ancestral, which is problematic, but it is different from what we see within the context of urbanization and industrialization. We see, for example, very recently, a description of at least 13 species of Segatella. Prevotella has been recently renamed into Segatella. And the Prevotella copri, now Segatella copri, is the one that's mostly found within the industrialized world. But outside of that, we actually have many other species of Segatella that actually colonize, and sometimes colonize by multiple species within the same gut. And we're losing that with urbanization and industrialization. So you can see this very nicely on the heat map over here, whereas you have westernized studies. This is a very, very large meta-analysis, and you can see that essentially you just have Segatella copri, the clade A, within the Western world, and then the non-Westernized populations, you have many, many different species of Segatella actually colonizing the gut. We can see this as general patterns as well. So the principal coordinate analysis here just pulls together many, many different studies and shows that you can see this transition from Westernized to transition into non-Westernized gut microbiomes. They are very, very intense, and this happens very early on since the colonization period during the first years of age, right? So this is actually a study that was looking at infants up to 3 years of age. We see changes, let's call them changes of lifestyle in humans, but we also see those in animals. And every time that we compare wild versus captive animals, we see changes in the gut microbiome. One of the hallmarks of urbanization and industrialization is the loss of diversity. And we have heard a lot about loss of diversity associated with disease. But even on health people, if you look at them, they have a lower diversity within the urbanized, westernized world than than we find in other countries. And animals are not different. So when we take animals that are kept in captivity, you tend to lose that diversity as well. We see that over and over in many different studies. This is another example here, which we can see very nicely within this panel, the separation between this carnivore group, which is very distinct between the captive versus the wild populations. So what we see is that changes have happened very quickly within human society. Within this last 200 years, let's round it to 200 years, we see the advent of the Industrial Revolution, which changes quite a bit how humans live. And everything that comes together with it, animals included, shifts together. This was enough time for us to shift to dietary patterns. We actually have the advantage of having antibiotics to treat disease. Nobody's saying that that's a bad thing. We are curing, diseases. That's a good thing. The problem is that something else got broken in the process. And now we're trying to re-understand this hygiene hypothesis of actually killing the bacteria and killing the pathogens, and that was really bad. And that was a very, very prevalent idea in the early 80s. And today we're reinterpreting that and actually saying, maybe it wasn't really the pathogen, but the fact that we killed everybody else too. And we're still killing everybody else. Not necessarily with antibiotics, but because of the changes that we're actually putting people through. So changes in diet, changes in how we deal with diet, and changes in the food matrix itself, and we heard about that here as well, do make a difference in how we colonize and how the microbiome actually behaves. So I'm going to present to you guys a little bit of the data that comes from my lab within this last years. Like I said, I'm very interested in populations, so when I got hired by University of São Paulo, my idea was to start characterizing populations, and then we had a pandemic, so we had some snags in the process. But we do have some data that I'll share with you, and we take advantage of some of the expertise within the department to actually do small trials to see how diet and different types of diet, different components of diet actually are influencing the gut microbiome. And occasionally with collaborators, we do into animal models, murine models as well. So I'll present some data to you guys about what we've been doing. As we saw very early on, carbohydrates seem to be one of those very important points of control of the gut microbiome. But not all carbohydrates are alike. We have seen very recently a very, very prominent paper that showed that we have lost the capacity within the human gut of actually digesting cellulose, that probably was something that we were very much capable to do to a very great extent, and we have lost those bacteria, even within non-westernized countries. We're very, very quickly losing that everywhere in the world. But everything else seems to still be digested to a certain extent. So I'll show first some data with beta-glucans, which was an intervention in a mouse model. And the idea here was to try to see what are the effects within the gut microbiome, if they were direct or indirect. How is it that they were dealing with this? So those are fungal beta-glucans. We actually did a series of gavages in these mice, and just did 16S sequencing on this small study to see what was happening here. Comparing two genotypes, deducting one knockout is actually the knockout for the receptor that actually is able to sense beta-glucans, and of course the wild-type mice is just the one that actually is able to completely recognize it. Beta-glucans, in this case, they're not from plant origin. Those are fungal beta-glucans. This is the major pathway that actually is present within the immune system to mount a defense against fungal infections. So, and we had some interesting initial data showing that there were some effects of this beta-glucan within, immune function within this mice. So we decided to take a look on gut microbiome. The first thing that we see is that there is a reduction in diversity within this gut of these animals. So it takes a little bit of time for the effect to actually be measurable. So what we have here is alpha diversity presented for the two genotypes over time. The T0 and T1 are prior to the gavage. And then the T2, we already have about 20 hours, 24 hours after the first gavage. And then after that, we see that there is this drop in diversity within the gut of these animals. We can see this very nicely represented here as beta diversity analysis. So depending on the molecule that actually coming in the diet, you can actually reduce the diversity quite significantly within the gut of these animals. We can put some names on some of those bacteria. Not all of them are very easily nameable because, ASVs for everybody that works with 16S. But we can see that even bacteria that are very prevalent, that all the mice within these cages actually had present, and this is the number of mice that had that bacteria, they basically go all below detection level. I'm not going to say that they were lost completely because we cannot say that, but they were reduced to a point that we can no longer detect them in the sequencing. So this, to us, it's sort of interesting because there is a lot of beta-glucan components actually coming into the market as products for people to consume. And what is that going to do to the gut microbiome of people that actually would like to have an immune modulating molecule within their guts? That's the selling point of those products coming into the market. So I think that we still have quite a bit to go to understand what is happening here in terms of maintaining and keeping up the diversity within our guts. The other piece of data that I'll present to you guys is a small clinical trial in humans, and I much prefer to work with humans, to be very fair. You can ask them several questions. You can actually interact with them in a more direct way. And what we decided to do was to compare in Brazil, in the context of Brazilian gut microbiome, what are the effects of resistant starch versus inulin. And those are two of the carbohydrates that we would use to actually act as fibers, right? Inulin very much used, and resistant starch studied, not so much used to this, commercially. This was the work that was done by Fabiana. She's currently now moved on from University of São Paulo. She's taken up a position at University of Limerick in Ireland. But the idea was to make a meal replacement for these people. It was delivered as soup, and they were consuming this for 6 weeks, and we just did the sequencing before and after the intervention, both with the two groups and with the placebo as well, and this was a double-blind study. This is submitted now, it's being evaluated, hopefully it will be published soon. So first things first, what we see in the gut microbiome of Brazilians, To this date, we don't have a lot of data. We're still very much underrepresented within the sequencing efforts. But the first thing that we see is that if you do the enterotyping analysis, as traditionally is done, we see a much larger prevalence of people that have a very clear Prevotella enterotype within their guts. So within the Western world, normally you would find about 10% of people that are really rich within their guts to have a lot of Prevotella. And we see about 40% of the people from São Paulo, in this case, that actually have a Prevotella type enterotype. And I like to emphasize that this is São Paulo people because really, a very large city, and we are a very localized study, so we are working with a very urban area. And I will present a little bit of data that actually follows up on these comparisons of different populations for you guys. What are the effects of the intervention? So we had inulin and resistant starch consumed, and what we see over here is just a representation of the different genes that actually are within the gut, functional potential of this microbiome, that was actually significantly modulated by the interventions. So on the bottom, the cluster 2 is the Prevotella cluster, and we see that the resistant starch consumed as the unripe banana flour has many different genes that actually all sorts of physiological pathways within those bacteria that actually are shifted and both upregulated and downregulated. And we have very modest effects of the inulin for the people that are Prevotella cluster. We see barely any changes within the Bacteroides people. So the background of the gut microbiome actually defines the effect that we have here. And I think that this is very much ignored quite often when we do think about interventions that are microbiome-centered. Very few people actually go out of their way to say, let's do something before we do an intervention and then decide what is it that we're going to use and apply. And I think that we should start to actually use this information. Some of the pathways that actually are modified are the propionate synthesis pathways. We can see several genes that are modified, but we also see some other interesting things. We see the entire lipopolysaccharide biosynthesis pathway-related genes that are all downregulated when we actually consume this type of fibers. So those effects are potentially very interesting also from the immune point. We did not do any analysis on immune function here, but we did look at gastrointestinal function. And very interestingly, we actually see that the inulin does affect function. Each one of the questions here that we are measuring, they come from a standardized intestinal function questionnaire. It's not really important which ones are the questions. Some of them deal with flatulence, some of them deal with transit time. And we see that the inulin is actually able to, change functioning. We just don't see those effects pronounced within the, the physiology of the bacteria within the gut. There is something else happening here on this context. And we see the same, we see some of the same effects also for the resistant starch intervention. So I think that we should start to really both for humans and perhaps for veterinary medicine as well, to put this into use before treatment. Perhaps even in veterinary medicine is already used to an extent with dysbiosis index, for example. But for humans, this is very much not the case. People usually do not have the kind of analysis done prior to any type of intervention. I'll finish presenting to you guys a comparison of two populations in Brazil. It's a follow-up study with the São Paulo population, and then we had the opportunity to collaborate with a colleague of mine who now is actually at the University of Pará in Brazil, in the north of Brazil, and he was preparing an expedition to go to the Amazon to do some bioanthropological work, and contact me, just say, let's do some gut microbiome studies here. This is great, we should do it. And I was like, sure. How do we do it? I was like, with great difficulty. So the location that actually was the study site in the Amazon is called Mamirauá. It is an agro-sustainable reservation in the middle of the Amazon. The closest city is actually 6 hours by boat from the closest local population. From the other side of the reservation, that is over 24 hours by boat. So We actually took liquid nitrogen to the field, like huge tanks, to actually be able to have a cold chain to sample the gut microbiome in the field and then bring it all the way down to São Paulo to study. This was an undertaking that I'm not sure we'll be able to repeat anytime soon. But we did that. And the figures here are just to present you what are those extremes. What is it that we're talking about when we're talking about those two populations? So Mamirawa, being an Amazonian rural area, is very much within the floodplain, so it's susceptible to the seasonality of the Amazon. You have a dry season and a wet season, and during the wet season, everything is very much flooded. And they are still very agrarian in the way of living. Their diet is very monotonous, and we do have very, very detailed characteristics of their diet. We got the data. I'll present a little bit to you. And in São Paulo, we have a very urban center, right? The largest urban center in South America, one of the largest ones in the world. And the figures just showed what São Paulo looks like, from the top, looking with your squinting eyes. And the idea was to see what is the difference between the ends of the spectrum. Because in Brazil, you find everything in between. You find the extremes, but you also find all the points in between this transition. And the transition happens not just in urbanization, but everything that comes with it. So we decided to start at the extremes and then, more recently moving into some of the in-betweens. So what do we see between these people? We're starting with alpha diversity. We see that São Paulo already has reduced alpha diversity when you compare it to the riverine people. So, and by all the metrics that are on the board, I particularly like the phylogenetic diversity here, but you can pick your phylogenetic measure. No matter how you look at it, we're losing bacteria within our gut already in São Paulo. Not always lost. There are still some things that are conserved. So if we look at the beta diversity within these populations, we see that by weighted and unweighted unifrac, we start to build up a picture that's a little bit more complete. So when you look at presence-absence data, unweighted unifrac, we can see that there is a nice separation between the two populations. So we really don't have some of the bacteria in one type of sample versus the other one. But when we started to put in the proportions of this bacteria into the equation and actually start to look into the weighted unifrac, we see that there is a lot of people in between, right? And we see that there is some people from the riverine community that's already starting to move towards the São Paulo pattern, but not always lost. We actually have a lot of people from São Paulo that still look very much like what we would find in the riverine population. So they haven't lost completely that profile of gut microbiome proportions that we see. Like I said, one of our interests is how diet modulates, the gut microbiome. And very, very early on, it became very clear to me that the matrix was important and how food was being delivered was actually important in the axis and what actually reached the colon, right, to be used. We heard about that yesterday as well. So we characterized the diets of these volunteers quite completely. We have nutritional composition, the traditional carbohydrates, macronutrients, micronutrients, but we also have a measure of processing for their diets. And we are able to actually establish, within some types of classifications, how processed the diet of a person is or how processed a type of food is. And what I'm showing to you here are all the different types of food groups that represent different types of sources of these carbohydrates from these two populations. There is no difference in the total amount of carbohydrates that they're eating. They still eat about the same amount of carbohydrates. The total energy intake between the two populations is the same. They don't really have a difference in the total amount of energy. There is a little bit of a difference on protein intake and lipid, right? Because the total energy is the same, but the macronutrients shifted a bit. To us, it was surprising that the protein intake is actually higher within the riverine population, a priori. Post hoc sort of makes sense when you think that they're fishermen, and they actually do fish quite a bit, and there are days on their recalls that we see fish being eaten 3 times a day because they had a very good catch and they kept eating the fish until it's over. You need to remember that they have no electricity. So all food that they consume needs to be shelf stable or needs to be consumed very quickly. Which poses a very interesting question for how Brazil is approaching food security. And the Brazilian government has created some programs over time to improve food security, but that doesn't come without consequences for the population that is using this. Because now they are being inserted within our market and are not consuming the foods that they usually traditionally would eat that are non-market foods. So they shifted, for example, to start eating wheat, which is not an Amazonian food, right? But flour is shelf-stable. They can go to the market with the monies that they do receive from the assistance programs and buy that, and that's shelf-stable, and take it back to their villages to then insert it within their diets. They also shifted the way that they're eating the fish. So traditionally, they have always eaten fish that was cooked as a stew, and they're shifting that to eating now fried fish because they can buy oil, which is also shelf-stable. So they bring oil back to the villages, and now they use this to actually fry the fish, which also lasts longer than the stew. So for them, it makes a lot of sense to do this shift in their diets, and this doesn't come without consequences to their health and to the microbiome. Just as an example, we see here that for the riverine, they still take most of their carbohydrate intake from cassava flour, which is one of their staple foods, and it has not changed. But we do see an insertion of several other types of foods. Some of them, for example, crackers, shelf-stable, foods, are starting to actually creep up within their diets. And in São Paulo, we see a pattern that's very distinct from that, with a much, much larger component of ultra-processed and processed foods as being part of their energy intake. If we zoom in a little bit more just in the fiber, we start to see some differences, right? So if we break those carbohydrates into different types of carbohydrates, then we do have a distinction in the amount of fiber that's consumed. Amazonians actually eat less fiber than São Paulo people. None of them eat any close to what would be the recommendation of 25 grams to 35 grams that we have today, but it is what they consume. We think it's actually pretty correct because what we see in São Paulo is close to the current national average, which has come down over this last 15, 20 years as Brazilians are shifting their diet overall. We see, again, the same type of pattern for the other macronutrients, and we're just wrapping up this story to actually be published. I'll finalize presenting the context of these populations, these two populations in Brazil, in relationship to all other populations that we could obtain from the literature that have compatible data. Not everything that's published is necessarily compatible, but we were able to take many, many different data sets from the literature and compare it to the São Paulo and the riverine community. So we see at the bottom of the graph that all the riverine people are right over here, very close to Yanomami, very close to several other non-industrialized, non-urbanized populations. And we see the São Paulo people in the steel color that's actually spread in the two sides of the graph. And on the top we have Americans and English people and very urban, very industrialized gut microbiome signatures. We would not be able to separate and distinguish those people from those signatures. But we do see a lot of Brazilians that are still within this side of the graph. So within the population, this hasn't really been lost yet. But it is slowly being reduced in frequency and occurrence within people. And if nothing happens, probably will be lost as well. I think that one of the points that we need to remember is that a lot of these studies are coming from the industrialized world, and a lot of people have started to say that we have to start to save the bacteria from the world because we're killing them together with everything else. And the implication is very simple, that eventually perhaps we should consider if we want to reinsert bacteria that have been lost within those guts. Whether that's going to be done or not is still up to debate. We don't have consensus at all on that, but there are calls that have been made and there are efforts that actually are happening right now to preserve the microbial diversity that we still have within our guts. The take-home messages here, I think, just to wrap up, are that we have a very long history of coevolution with our gut microbiomes. Not just us, but every single animal that we can think of. That has been shifted quite drastically by how we shift our lifestyles. So, and this has consequences. This has consequences to health. This has consequences for the future. And if we are to use the microbiome, we should start to use the information they already have to start to integrate it within these practices. For example, as prior knowledge before you do an intervention, right? So that we can really do more targeted interventions, especially when we're talking about nutrient-based interventions, but probably for all types of interventions that actually deal with the gut microbiome. I'll finish just thanking the people in my lab and the funding source. Always nice to thank the people with the money because, you know, I'd like to have more money to do more studies. And the people that actually does the magic within the lab, I couldn't have done all of this without them. So with that, I finished a little bit early, which is great because then we can go to have some wine. Thank you so much, and I'll take some questions now.